works for you. Is it okay?
Yeah, I'll get it going now. Okay, is that working?
Well, almost. Yes, we see already. Okay, very good. So, dear listeners, dear colleagues, it's a pleasure for me to introduce our today's speaker, Professor Moore from Imperial College in London. And Professor Moore will tell us about, well, here it is written lymphatic system biomechanics. In the announcement we had it's a little bit different, biotransport mechanisms in adaptive immunity. Anyway, Professor Moore, you are very welcome, and we are listening to you.
Thank you. It's a great honor and a pleasure to be with you today and tell you about some of our research in the lymphatic system. So on the left is a list of the different projects that we have in the laboratory, and you can see the ones that are darker have to do with the lymphatic system, and that's what I'll be focusing on today. There are some other projects going on in the lab, and I'm also involved in medical device design and entrepreneurship. So we try to learn from our research and establish new techniques to help patients suffering from diseases such as lymphedema and cancer, and then also we look at problems associated with immunity, which is going to be the main focus of most of what I will say today. So in my laboratory, we do some experiments, and you'll see that. We do some modeling, some computational modeling, and I know that'll be a great interest to the mathematicians. And then the device development that I mentioned earlier. So we, for example, are developing some devices to help treat lymphedema and help patients suffering from that incurable disease. So the lymphatic system is a very interesting sort of third part to the circulation. It pumps fluid around the body, but in a very different way from the blood system. So when your body pumps blood around, it goes through capillaries, these very small tubes where gas exchange takes place. And at the capillary level, there is a net flow of fluid out into the interstitial space. And that fluid needs to be gathered up and returned to the blood system. Otherwise, you end up with swollen tissues. And lymphedema is what we call the disease when the tissues swell due to a breakdown in the lymphatic system. So the lymphatic system's job is to take in this fluid and pump it through at least one lymph node on the way back, and I'll spend a fair amount of time talking about that. And then eventually all of that fluid comes back into the blood system. So these lymph nodes are an important means of monitoring the fluid coming in. So the fluid coming in may have dirt and debris, cancer cells. There are a lot of things that can come into the initial lymphatics. And that fluid needs to be cleaned before it goes back into the body. Now, in terms of numbers, you're probably familiar with these numbers of cardiac output, blood pressure, and then the aortic diameter. So the diameter of the largest blood vessel in the body is 2 to 3 centimeters in the adult human. In the lymphatic system, you can see we only pump about 5 liters per day. So much lower flow rate than the blood vessels, much lower pressure. So some lymphatic vessels operate at sub-atmospheric pressures, and that's a natural part of their function, and they do it very well. It's a very interesting phenomenon how they can continue to pump in the presence of sub-atmospheric pressures, given how they pump. The thoracic duct is the largest lymphatic vessel in the body, and it's about 2 to 3 millimeters in diameter. So the largest lymphatic vessel is an order of magnitude smaller than the largest blood vessel. So the lymphatic vessels are low flow, low pressure, very small diameters, And the fluid is essentially clear. It's water with some proteins and some cells in it. And so the lymphatic system is actually very difficult to observe. So if a surgeon cuts into the body, undoubtedly they're cutting through many lymphatic vessels. But because the fluid is clear, the vessels are small, they're very hard to see, they may never know that they cut through lymphatic vessels. So the history of lymphatic research is not nearly as populated with knowledge as the blood flow system. So that's one of the challenges that we have to overcome in the lymphatic system, is there's just not that much known about it. Now, what I'm going to tell you today is a story about adaptive immunity. And it's important, the theme that's going to run through this, the important theme that will run through this is that adaptive immunity relies on fluid flow. So this, you can think of this as a flow-mediated information delivery system. So for example, here we have a vaccine. It's got some antigen and some adjuvant in it. I'll come back to tell you what those are later on. The antigen is the information on the COVID virus, for example, that is to be fought off later on. Now this gets injected into the intermuscular space here, IM. There's also the possibility of injecting vaccines in the subcutaneous space or intradermal space. But regardless of where it goes in, that information needs to be taken into these lymphatic vessels located in these different spaces. And you can see there are different densities of lymphatic vessels in those different spaces. Now, the thing that transports that information into the lymphatic system is interstitial flow. So the flow of fluid in between the body's tissues. So that is how the information gets gathered into the lymphatic system, is via this very slow interstitial flow, kind of like the flow of water or oil in the rocks of the earth. Now, as the antigens, these things that might hurt you, they can flow in solution as just suspended as a solute, or they might be captured by cells, these antigen-presenting cells, that live in the skin, as you can see here with these dendritic cells, or other cells that sort of sit around here and wait for bad things to come in. So these antigen-presenting cells or the antigen will then get transported into the lymphatic system. And then the larger lymphatics, the ones that you see depicted here, do the active pumping to transport that information to the lymph node. Now, the lymph node, you have about 500 or 600 lymph nodes in your body. And you can think of this as an information exchange organ. So the information that is coming in here needs to be transferred to either T cells here in the middle, or B cells, which are collected sort of around the outside of the lymph node. And that information must be delivered very precisely to the right kinds of cells in sufficient concentrations to form adaptive immunity. In other words, to teach your body's immune cells that there is something bad here and you need to learn about it so you can fight it off. So that fluid flow guides that information delivery either through direct delivery of the antigens that are in solution or through guiding the antigen-presenting cells. Now, that's actually what I'm going to talk about today, is the lymph flow inside the lymph node, how that distributes the antigens and helps guide the antigen-presenting cells within the lymph node. Then I'll come back to talk about the pumping of these larger lymphatic vessels here that have the job of delivering the fluid from the periphery to the lymph node. And then at the end, I'll come back and talk about how that plays a role in adaptive immunity. So that's the plan for today. And by the way, at any time, if you have questions, please don't hesitate to interrupt me. I can't see the chat, so if you want to post questions in the chat, I'll maybe check that at the end, but if please just interrupt me and I'm happy to answer any questions you have. So what happens when fluid comes into the lymph node? Well, each lymph node typically has multiple incoming or afferent lymphatic vessels. So that fluid flow comes in here, and it might go directly into the B cell region here. It might go directly into the T cell region, or it might go around the edge through what's known as the subcapsular sinus. Now, the subcapsular sinus is a relatively open flow region, so fluid flows very easily through the subcapsular sinus compared to the densely packed porous medium region that forms the rest of the lymph node. So the fluid can either flow in the subcapsular sinus and around directly to the medullary sinus, which is another sort of loosely packed region that collects fluid so that it can go out the exiting lymphatic vessel or the efferent lymphatic vessel. So there are multiple paths that the fluid can take as it goes through the lymph node. It's also important to realize that there are blood vessels distributed through the lymph node here, and that fluid can move into the blood vessels or out of the blood vessels, either way. It depends on the local flow conditions, and I'll come back to that later on. So this is what flow looks like in a lymph node. This is the result of a computational model, so CFD. And so we are calculating flow in the subcapsular sinus with the Navier-Stokes equations. And we are calculating flow in the porous medium region here in the middle with a combination of Brinkman and Darcy flow. So these are large collections of partial differential equations. And what we find is that almost all of the flow follows the path of least resistance. So the fluid flows around the edge, the subcapsular sinus, and then down into the medullary sinus, and only about 10% of the flow actually goes into the middle of the lymph node where the immune cells are. So that surprised us. But then one thing you have to remember is that this is under normal conditions. When your body is healthy, there are no antigens to respond to, then only a small portion of the fluid needs to be monitored for antigens. Otherwise, the rest goes around the edge in a very low resistance pathway so that the fluid does not back up and you develop edema. Now, these flow patterns here are, as I mentioned earlier, important for the delivery of antigens that are suspended in the fluid. And there are multiple pathways for that to happen. I won't spend much time talking about that. But then what I will spend some time telling you about is that these flow patterns are important for setting up concentration gradients of substances known as chemokines that direct the migration of antigen-presenting cells. Now, how do these flow patterns change when we get a vaccination? So we've done this experiment in mice. So we inject a vaccine into the foot of the mouse, and then we look at what happens inside one of the mouse's lymph nodes here in the hind limb. And here are some experimental details on how we do that. That's what the lymph node looks like in the leg. Mouse lymph nodes are about 1 millimeter in diameter, so that's about an order of magnitude smaller than human lymph nodes. But we can take these lymph nodes out and we can cannulate them. And I'll tell you, this is a very difficult experiment because the incoming and outgoing lymphatic vessels are only about 50 to 70 microns in diameter. That is about half the diameter of a human hair. So it's very difficult to cannulate or insert a tube inside these vessels, but we can do that. And then we pump the flow through the lymph node, and we measure the amount of flow and the amount of pressure difference necessary to drive that flow. And that tells us about the flow resistance of the lymph node. And keep in mind, the flow rates in these vessels are on the order of microliters per hour. And the pressure differences are in the order of maybe a couple of centimeters of water. So these are very exquisite experiments that require special equipment to measure such small quantities of flow and pressure. So this is what we find with these lymph nodes when we activate them with TLR. So this is a vaccine adjuvant that works well in mice to accelerate the response of the immune system. And what we find with these nodes is that they swell up. They get much larger in volume by a factor of about 5, well, about 3 in this case. And at 72 hours, the lymph nodes are still enlarged. Now, you may know this already. You may know that swollen lymph nodes in your neck, for example, can be a sign that your body is responding to some kind of infection. And that is what we find with these mice. So lymph node swelling is nothing new. It's something that we expected to see in this experiment, and sure enough we do. And here are some pictures of what those lymph nodes look like. So these would be the normal lymph nodes over here, and these are the ones that have received the vaccination. So you can see they're quite swollen, they're much heavier and much larger. Now, when we measured the flow resistance across the lymph node, and again, that involves measuring the pressure drop across the lymph node as well as the flow rate, what we find is that the flow resistance of those lymph nodes goes up by about an order of magnitude within 24 hours. But then after 72 hours, the flow resistance has come back down to its baseline level. And what I'll remind you is that the lymph node is still swollen at 72 hours. So it's adjusted its flow resistance probably as part of the swelling process within the first 24 hours. But then after 72 hours, the lymph node is still very large, but its flow resistance has returned to the baseline level. So this puzzled us very much, and we wanted to know, in particular, what part of the lymph node changes to increase the flow resistance. So we take these experimental results and we go back to our computational model. Remember, the lymph node is only about 1 millimeter in diameter. It's impossible to tell within such a small lymph node what has changed inside the lymph node to create that change in resistance. So we go back to our computational model and we start changing things. So one thing we can change is the hydraulic conductivity, or the porosity, you might say, of the T cell cortex, or this big porous medium region in the middle here. Now, remember that 90% of the flow goes around the edge in normal conditions, and you can vary the conductivity of this T cell cortex all you want. It makes no difference at all. So that is not what is causing the change in flow resistance. Can we change the height of the subcapsular sinus and explain the change in flow resistance? So remember, the subcapsular sinus is that open channel region around the edge that normally forms the low resistance pathway. So we took the subcapsular sinus height, which was 20 microns in the baseline case. We changed it to 10, and it made almost no difference on the overall flow resistance of the lymph node. And remember, this is the flow resistance of the lymph node. This is the flow resistance after 24 hours, and we're trying to imitate that experiment with our computational model. The next thing we did was we changed the hydraulic conductivity, or the porosity, of the medullary sinus, or this exit sort of fluid collecting region that leads to the outgoing lymphatic vessel. So we changed that by an order of magnitude, which is not difficult to do. If you're familiar with porous medium flow, you know that you can change the hydraulic conductivity just by a small contraction of the volume, and it changes the hydraulic conductivity by an order of magnitude easily. So that does actually explain very well the change in overall flow resistance that we measured in the experiment. And we tried many other things. We tried changing different parts of the lymph node. We made it larger. There was nothing that we could change about the lymph node except the hydraulic conductivity of the medullary sinus that allowed us to explain, that reproduced the experimental result. So we think there must be some special cell type in the medullary sinus that is responsible for changing that flow resistance.
Excuse me, Professor Moore, if I can ask a question about this part.
Yes.
So I'm a little bit surprised that when you say that you change the diameter of this, like H, what is written here, H 20 micrometers of this part of these vessels, and this does not really influence the hydraulic resistance, because just like intuitively we can imagine if you have just a pipe and if you decrease twice the diameter of this pipe, then like the resistance will be twice larger. So why it doesn't work like that here?
So you're correct. The resistance did increase, but not as much as we saw in the experiment. So it did increase the overall flow resistance of the lymph node, but not by very much. So you are correct that if we only measured the resistance of the subcapsular sinus, then we would probably measure, as you say, about a doubling or 4x, maybe the flow resistance, by cutting the height in half. But remember, the only resistance we can measure is the entire flow resistance across the entire lymph node. And it turns out this is a fairly low resistance value in parallel and in series with much higher resistance values. So changing a low resistor that's in series, and in parallel with other resistors doesn't make that much of a difference in the overall resistance.
Okay, thanks.
Yep, no problem. So then we look at how that change in flow resistance affects the amount of fluid that's coming into the central part of the lymph node. And sure enough, when we change the hydraulic conductivity of the medullary sinus from this vertical line to this vertical line, it changes the amount of flow that goes into the T cell region by about 3x. So now that the low resistance flow pathway around the edge of the node has been shut down, there's more flow coming into the middle part of the lymph node where you have your T cells and your B cells, or the cells that need to learn about the important antigen information coming in. So that all makes sense that the lymph node has developed a way to shunt more of the flow into the middle of the lymph node. Now, if you recall, I told you there are blood vessels on the inside of the lymph node here. And what we've done in the work that I've shown you so far is a very simple thing. We've just assumed that the blood, the fluid flow that goes across the blood vessel wall is dependent on the difference between the lymph pressure in the lymph node and the blood pressure. And then also the oncotic pressure. It's related to, it's osmotic pressure specific to these proteins in the body. So there are pressure differences driving the flow, and then there's a hydraulic conductivity and a surface area of the blood vessels that determines how much flow moves across. So what I've shown you so far is a very simple, homogeneous model of the lymph node in which we have assumed that the blood vessels are uniformly distributed inside the lymph nodes, so that the blood vessel surface area per unit volume is continually distributed and uniformly distributed amongst the entire lymph node. I've also made an assumption in that model that the blood pressure is uniform inside the lymph nodes, so that the driving pressure here, this component of the driving pressure, is spatially constant. And we know this is not true. Blood vessels are not uniformly distributed generally in any tissue. The pressure in arteries is much higher than the pressure in veins. And so this is something that we decided we needed to know, because that first model of the lymph node told us something we didn't know about the lymph nodes. But we're not sure we trust that aspect of the model, and we need to know more information about it. And the way we can get that would be to do imaging of the lymph node blood vessels. In other words, the specialized blood vessels that are inside the lymph nodes. So that's what we did. This is from micro-CT. Again, this is a mouse lymph node. This is about 1 millimeter in diameter. And you can see that these vessels are color-coded by diameter. So one of these large red vessels is an artery. The other large red vessel is a vein. And those distribute blood throughout the lymph node. And the blue vessels are the capillaries where the fluid flow, fluid exchange takes place and the gas exchange takes place. Now, once we have the geometry, then we can simulate computationally the blood flow through this system and determine the distribution of blood pressure. So again, the pressure is going to be higher in the arteries than it is in the vein. But we can also estimate the distribution of surface area. And remember, that's important for the fluid exchange with the lymphatic compartment inside the lymph node. So these maps show you how those the blood vessel surface area is distributed throughout the lymph node. And these are 4 different lymph nodes that we have measured here. So the surface area density, as you can see, is microns squared over microns cubed. So that's area per volume, if you think of it that way. And what you will see is that the surface area is distributed around the outside of the lymph node. And that was a consistent finding in all the lymph nodes we looked at. So the blood vessels are not uniformly distributed. It seems they're purposefully distributed around the outside. And to this day, we don't know why that is. But at least now we have the information, and we can incorporate that into our computational model of fluid flow through the lymph node. And when we do that, so this first block here is the blood vessel pressure. So that is the pressure that we get from calculating blood flow through that complex arterial and venous tree that I showed you earlier. And you can see the highest pressures are where the arteries are coming in, where the veins are coming in. Oh, sorry, the arteries coming in. And this is how the surface area is distributed, again, mostly around the periphery. And it turns out that the surface area distribution is by far the dominating factor in determining how much fluid moves across the blood vessel wall. So this is not the flow of the blood. This is not the flow of the lymph. This is the flow of the fluid in the blood vessel wall from the lymph compartment to the blood compartment. And you can see that's mainly following the pattern of where the surface area is. So there's some histograms down here of these different variables. And another thing that we can do in the computational model is, of course, change anything we want. So in this case, we changed the lymph pressure from very low to very high. And we see that the movement of fluid across all of the blood vessels added together is probably normally going to be from the lymph compartment to the blood compartment. But then there's a small area down here where we can see fluid moving from the blood compartment to the lymph compartment. And we think that's important for lymph node swelling early in the response to vaccines, as I showed you earlier.
Excuse me if I can interrupt
Sure.
you again.
Yeah.
How do you do the simulations? Do you consider individual vessels, or you can do some kind of averaging or porous medium approach or something else?
So the blood vessels, we calculate the flow in every single blood vessel.
Oh, but
use,
there are many of them.
Yeah, that's true. But we do, it's a simple Poiseuille flow based model. Okay. And actually, we've also incorporated an apparent viscosity to incorporate, in some sense, the non-Newtonian behavior of blood. But we calculate, yes, we do manage to calculate blood flow in each one of these tiny little blood vessels, but we assume Poiseuille flow. So it's not full 3D CFD.
But how many vessels are here? It looks like thousands of them.
Yeah, it is. It is. So you can see in the histograms here.
Oh yeah.
Yeah.
Well,
so that's, it's thousands. Yeah.
I would say poor graduate students who do that.
Yeah. Well, he was a very determined graduate student. We need good, determined graduate students, don't we? So yeah, but the blood vessel flow is calculated as I described, but yes, we are still using porous medium flow to describe how the lymph flow moves around the blood vessels, and that this calculation is moving from the lymph compartment into the blood vessels. So now, what does that flow have to do with guiding dendritic cell or antigen-presenting cell migration within the lymph node? Well, that has to do with chemokines. So chemokines are cytokines that are specific to immune cells. So cytokines basically stimulate cells to move from one direction from one location to another in a preferential way. And it's something like when you walk past a bakery and you smell the bread baking, you want to walk towards the bakery, and the smell of the bread gets stronger, and it makes you hungry and makes you want to go inside to buy the bread, right? So cells do the same thing. They're able to detect where the stronger concentration of the chemokines are, and they move in that direction. So I won't get bogged down in the terminology, but we're interested in these two different chemokines, CCL19 and CCL21. They are important for guiding the dendritic cells that have captured antigen and need to present it to the correct cells within the lymph node. So that's what we would like to study, is the transport of these chemokines within the lymph node and how they set up these concentration gradients to guide the cell migration within the lymph node. So CCL19 and CCL21 are similar, although the CCL21 will bind to extracellular matrix, whereas CCL19 does not. But they are both produced by certain cell types, and they can diffuse throughout the lymph node. They can also be infected by fluid flow. They can bind to receptors on the cells, and there are all sorts of interesting biology in terms of how these chemokines interact with the cells around them. But you can see it's quite a complex system to try to model. But I'll show you now, since this is a maths department, you won't be afraid of the equations. I would never do this to a biologist, but I'll show the equations that we use to calculate the chemokine concentrations inside the lymph node. So here's the fluid flow velocity. That is what I showed you earlier in our lymph node flow model. So we calculate that using CFD, and we use it as an input into the mass transport modeling. In each row, you will see the first 4 rows contain a different, first 3 rows, a different representation of chemokine. So chemokine can be floating freely in the fluid. It can be bound to the extracellular matrix, or the sort of gum that is between the cells of your tissues. It can be cleaved, which is a way of saying it's been chemically modified by a cell. It can bind to dendritic cells, and the cells then are represented as having bound to the chemokine. And so these last 4 equations here are different cell populations. So here we have fusion and infection. So go ahead, sorry.
Sorry, no, maybe it's what you are going to say, but I see this in the last equations, like the third from the bottom. There is nonlinear diffusion, right? Or what is that? This.
That's right. Yeah.
This. You are going to say it's chemotaxis, which is.
That's right. It is. Yes. Okay. Very good. You have a good eye. So
it's a little bit small, but okay.
Yeah. So you can see mass being conserved. So as unbound chemokine binds, it shows up over here, and vice versa. And then cleaving as well, and we can also lose chemokine. It can be transported away or decay, and that's the migration term. So yes, you are correct. So that's we do express the strength of the guidance with this term chi, which it depends on the bound chemokine concentration, which is this one up here. And then the cells can also just walk around randomly, which is represented by this. But this is the guided migration term here. So this is what the chemokine concentrations look like inside the lymph node, taking all of that into account. So these are unbound CCL19 concentrations down this column. This is unbound CCL21. And because CCL21 binds, but CCL19 does not, we also show a plot of bound CCL21 here in this column. Now, these four cases are sort of, this is the wild type, you might say, or where we include the flow and the alternate receptors that can take up the chemokine. This is without flow. You can see that not much changes if all you do is take out the flow. Here is if we take out the chemokine receptors that can just scavenge the chemokine. And this is the case where we knock out both the flow and the receptors that take up chemokine. And now you see something very different here. Now, the reason we did this is because there was a very well-known paper in the field that showed that knocking out this receptor in mice, which is something you can do biologically, made a big difference on the chemokine concentrations. And we were puzzled by that because, when we ran this model, we thought, well, there's no real difference here between these two. But what happened was when we ran the flow knockout case with the biological knockout, we found a big difference and explained their result. And what we looked back in their methods, and what they had done was they had harvested the lymph nodes and taken 3 or 4 hours to process them, during which time there was no flow going through the lymph node. So they were biologists. They didn't know that they had knocked out the flow as well, and they didn't take that into account. But we were able to explain it. And in particular, what is of interest here is this concentration gradient of CCL21 in this zone between the two B cell zones here. So if you look at how that varies, and it's just this plot here. So this is the bound CCL21 on the vertical axis and distance from the subcapsular sinus, so going inward in the direction of these red arrows. You can see the wild type case in the solid line here. If you knock out just the flow, it changes a little bit. If you knock out ACKR4, it does nothing. But if you knock out both the flow and the ACKR4, then you get rid of the concentration gradient. So this is like the dendritic cell walking past the bakery. It smells the stronger and stronger smell as it goes in there. But when you make that a flat concentration field, the dendritic cell doesn't know where to go, and that interrupts the immune cell process. So what I've told you so far is that under baseline conditions, most of the flow goes around the low resistance pathway in the lymph node. When you get a vaccination, that strongly shifts the fluid from the going around that low resistance pathway into the paracortex. The flow patterns do depend on the blood vessel network geometry, and we know we need to take that into account. And then what I've just shown you here is that the flow patterns shape the chemokine gradients that guide antigen-presenting cells within the lymph node. So I will then shift back to this picture of the vaccine. So we've got the antigen, the thing that can harm you, mixed with something called an adjuvant. And the idea with the adjuvant is to enhance the immune system's response. So I'd like to talk about that a little bit in this little bit of time I have remaining here. And so we got interested in adjuvants because they have sort of a strange history. Like many discoveries in medicine, we'd like to say in London, they start in London. So this is a set of experiments that occurred in London. So a man named Alexander Glennie noticed that vaccines in his animals worked better when the animals showed signs of inflammation. So he started adding aluminum salt, or alum, to the vaccine and found that it reliably produced information. It increased the efficacy of the vaccine, and away they went. There was no human testing. There was no further animal testing. They just started using it in vaccines around the world. Now, this is almost 100 years ago, right? We have regulatory systems in place that would stop this now, but aluminum salts have been used in millions and millions of vaccines. Every single one of us has had aluminum salts injected into us as part of a vaccine. So the question is, how does it really work? If you think about that transport system from the injection spot into the lymph node, what part of that pathway is affected by the vaccine adjuvant? Glennie himself proposed something called the depot theory, which is that at the injection site, the adjuvant kicks up the response, attracts immune cells there, those dendritic cells, to take up the antigen, and then magic happens downstream, I suppose. That was all he thought about. And he had no evidence for this, but this today is still what people believe. There's no real experimental support for the depot theory, but people still believe it. And since then, there have been some other types of adjuvant developed, including the ones we used in the experiments I showed you earlier. But really, no one knows how they work either. No one knows if they work in the injection site or in the lymph node. And I think it's time we figured out how these things work. We've been using them for 100 years, and we'll certainly do better than randomly guess how to use adjuvants. So I'm going to accelerate a bit because I want to leave enough time for questions at the end here. But let's just quickly talk timescales. So we know that when you get a vaccination, the information shows up in your lymph node within minutes. So how does it get there? Well, cell motility. Well, the biologists think that antigen-presenting cells crawl from wherever they picked up the antigen into the lymph node. And their crawling velocity has been measured at about 5 nanometers per minute. And if you convert that up into time and space, then you can see that it would take 4 years for that cell to crawl a distance of 1 centimeter. So getting from your shoulder into your lymph nodes does not happen because the cells crawl there. It happens because they get pumped there by the lymphatic vessels. And this is what that looks like. So these are immune cells flowing through a lymphatic vessel. They're flowing at a velocity of about 1 centimeter per second. So they're getting to the lymph node much faster than if they had to crawl there. So you can see that the lymphatic vessel is contracting. You can see another lymphatic vessel over here. This is an experiment where the vessel has been taken out of the body. So these vessels squeeze, and there are check valves to keep flow moving in the right direction. Now, we've spent a lot of time in my lab trying to understand this pumping of the lymphatic system and characterizing it. And I won't spend much time on it because I want to finish the talk and have time for questions. But we've constructed mathematical models of these lymphatic vessels pumping, and you can see they squeeze, about every 2 or 3 seconds, and they generate enough flow to keep your body from swelling up. So what we find with these vaccine adjuvants is that they downregulate lymphatic pumping. So this is alum salts. This is what Glennie discovered. And these are the 2 other types of vaccine adjuvants. So this is an oil emulsion, and this is a TLR activator. So you can see the vessels are told to relax by the vaccine adjuvant, and the vessels are told to contract less frequently. So it slows down the lymphatic pumping when you add an adjuvant into a vaccine. And this puzzles us. We haven't published this yet. This is brand new information. We don't know why this is. And given this, why do adjuvants even help? If they slow down the delivery of information into the lymph node, how do they help develop adaptive immunity? So you can see there's a lot we need to understand about vaccine information delivery. and the role of fluid mechanics. So this is where vaccines are delivered. So if you've had a coronavirus vaccine, it's gone into the intramuscular space where there are relatively few lymphatics compared to injecting vaccines into the subcutaneous space or the intradermal space where there are many more lymphatics. So is that the right strategy? Well, I'm not sure we know. People are trying injections into the intradermal space, and it seems like it gets the information to the lymph node faster. So that needs to be investigated. Transport through prenodal vessels, or these pumping lymphatics that I showed you, so that is down-regulated. It slows, the adjuvants slow down the flow into the lymph node. And then they certainly change the way the flow is going into the lymph node once they get there. So they shut down, the adjuvants shut down the low resistance pathway, and they send more information into the middle here, which is something that you can imagine does help adaptive immunity. So this part makes sense. This part does not make sense, I don't think, but we will find out. I think the right way to think about this is to start with the lymph node. So what is the temporal profile of antigen information delivery required by the lymph node to optimize adaptive immunity? If we can find that out, then we can design adjuvants that provide that profile that the lymph node needs. So wrapping up, lymphatics, it's a mysterious system, you might say. It's been long ignored in medicine. We have Professor Bocharov and other experts scattered around the world in a very few places that are investigating this system and doing all we can to learn more about it. Lymphatic pumping is crucial, as you can see, for delivering information into the lymph node in a reasonable time frame. And with regard to vaccines, again, what does the lymph node need and how do we deliver it? Adjuvants have never been looked at this way. And we want to do that. We want to help improve vaccines. And obviously, we all see today the importance of vaccines in saving lives around the world. So we want to make them better. So here is my contact information. Here is my website. If you'd have any other questions or would like to see some of our papers, I'm happy to send them to you. And I would like to just close with some words of thanks to many people who have helped out with this research. Some of them my PhD students that have gone on to other places, and collaborators from around the world and funding sources. And with that, I thank you very much for your attention and for your scientific fellowship, and I look forward to answering your questions.
Thank you very much for your very interesting presentation. We are really very excited about all these works, also ongoing works. It's really amazing the results you obtain. So please, I address to our audience, if there are questions, please, we have time to ask questions and to discuss.
May I ask a question?
Yes, yes, yeah. Please.
Thank you. Thank you very much, James, for your outstanding talk. Very inspiring. I have the following question to you. Concerning the information transfer in a lymph node, how would you rank the contribution of the conduit system as far as the delivery of signaling molecules into the internal domains of the lymph node is considered?
It's hugely important, and I love the work that you've done with your collaborators in Switzerland and otherwise on that. It's a beautiful system. So the conduits he's referring to are these very small collagen bundles that traverse through the entire lymph node, and they transport very small antigens directly into the T cell region, the B cell region, and so that system is hugely important. It's not something that we've taken into account in anything that I've shown you today, but we are working to characterize not only the geometry of the conduit system, but also the transport. And I think there's certainly a lot the world doesn't know about the conduit system. What we do know is just scratching the surface. But yes, its network structure, as you've investigated, I think is hugely interesting and very, very important. You know, you would like to think that the system has optimized itself over millions of years to deliver that information. I look forward to finding out if that's the case someday, hopefully.
Yeah. Yes. Yes. And I mean, maybe just I mean, a second question. With the facilities that you have, do you actually work on artificial lymph nodes?
So we are developing an implantable device to replace lymph nodes in breast cancer patients who have undergone lymph node dissection. So when you take lymph nodes out of the body, and for breast cancer patients, they're removing lymph nodes from under the arm here to help prevent the spread of cancer. That disrupts the drainage pathway out of the arm, and about 20% of all breast cancer patients end up with a swollen arm. It's a terrible disease. So I didn't talk about that today, but yes, we are developing an implant that would restore the fluid flow that's been disrupted and hopefully prevent these patients from getting lymphedema. Yeah, I've met these patients, and it absolutely wrecks their lives. It's a terrible disease, and I hope we can help them. We're not trying to replace immune function with these implants yet. That's something, we just want to keep the fluid moving, but that's something I think we can look forward to in maybe a decade or something when we get these devices out into the clinic. Then we can start thinking about immune function.
James, and just the last question, if I may. There is a highly organized network of the fibroblastic reticular cells, which actually guide the migration of cells within the lymph nodes, and in this way contribute to the information delivery and the response of cells to the chemokine gradients via. It could be, haptotaxis as well, chemotaxis. Do you also plan to incorporate that in your model?
So we have incorporated their production of chemokines in our chemokine transport model, but in a very a stupid way, you might say. We're assuming they're uniformly distributed, and we know that's not the case. So I think we would like to increase the spatial resolution and the physiologic fidelity of our model, and again, the type of network analysis you do is tremendously helpful in those efforts.
Thank you very much again, James.
Oh, thank you. It's a pleasure to see you again.
Yes, please. Other questions? So you mentioned that this adjuvant increases the immune response. So how important there is? What is the difference? Is it really big or not so much?
For some vaccines, it's the key to their success. For some vaccines, it doesn't matter. So the vaccine that I received for coronavirus does not have an adjuvant in it. Others do. And if you ask a vaccine designer how they choose an adjuvant, most of them will admit that it's a random process. It's almost alchemy. They just think, okay, well, we used this adjuvant in the last vaccine. Let's try it for this one. There's very little science behind it, actually.
And then you say that one of the strange effects that it decreases pumping in the lymph vessels. So is it also this difference important, or in the flow velocity, in the lymph flow velocity?
Yeah, we don't know yet. So we've written I think 4 proposals now to get that work funded, and we've not been successful yet. I think I love biologists. I work with biologists, but they don't understand fluid flow. They still think cells have to crawl to a lymph node, and they don't understand why should they give money to look at fluid flow, but we're still trying. So I hope to maybe someday I'll come to Moscow and visit and
excellent.
We'll talk about that story.
We fix our next seminar, and you will tell us about this new work. Yes, very good. And then also a very mysterious question for me, because in fact I knew about this lymphatic system from Annastacia, and she explained that, as you also mentioned, that little is known about that and also how it works. And in the beginning of your talk, you showed this, or maybe not in the beginning, you showed this small video where with this lymphatic system, how just it pushes, how it pumping the fluid, but how it works, what is the mechanism? Is it vessel walls which contract and do this thing, or what it is?
Yeah, so it's not peristaltic flow like your intestines. You know, they gradually squeeze like this to move the food in your intestines.
Yeah.
So that's not that's not how lymphatic. So they contract uniformly.
Uniformly.
And then they have the check valves that keep the flow moving in the right
But I suppose uniformly just a section, just a part of it.
That's right.
And then one part after another, right? It's like, yeah.
Yes, well, it's an interesting phenomenon actually. So there's a segment that squeezes, and then there's a valve, and then there's another segment. And you would think it would make sense. This one squeezes, then this one, right? And it's best if they squeeze out of phase with each other. Sometimes we see that. Sometimes they squeeze together. Sometimes this one will squeeze and this one does nothing, and it's very noisy and definitely not optimized. There's nothing efficient about the lymphatic system, and I don't know why.
So you think it's really occasionally like that, or there is a clever mechanism behind which makes it like that?
I think the lymphatic system relies on a lot of external tissue movement. So if you squeeze my arm, then you're contracting a bunch of lymphatic vessels and sending fluid forward. And I think so the valves need to be efficient. And that's what we find. They are mostly very efficient valves, but the squeezing part of the pumping is not particularly efficient. Every time we look for some sign of optimization in the frequency or the phase, or and we find nothing optimal about it.
Yes, this is very interesting, because we often think that all these physiological systems are very optimal, but apparently not, right?
Well, it's good enough to do its job, I think, is my conclusion of it. And the external tissue movement is free pumping, right? So if I squeeze my muscle, I get free pumping for the lymphatic system, and I'm squeezing muscle for other reasons. So,
but sorry, maybe you answered, but maybe I just lost something. This squeezing is due to muscles, or vessel walls can squeeze themselves?
They can also squeeze themselves. Yes, with very specialized muscle cells in the walls.
Yeah. Okay. Very good.
So I think Igor had a question. Igor is good.
Yeah, good to see you too. Brilliant talk as always. Thank you.
Oh, thank you.
I just was intrigued by you described the vascular system quite non-uniform and with some kind of almost like a cavity, or the strong gradient of the density of vessels. Is it right? In the lymph nodes, where you have the less density in the center and high in the periphery.
Yeah, the blood vessels, right?
Yes.
So those are the blood vessels, not the lymphatic vessels. So if I maybe share my screen again. So those vessels, right? And then they are distributed in the lymph node. So that's the distribution of surface area within the lymph node. So the most of the surface area, as you can imagine, is in the capillaries, just because there are so many of them. And that's roughly representative of where the capillaries are in the lymph node.
Does it reflect the density of the porous medium, or you expect it to be?
We don't know. We don't know. There's not enough good quality imaging of lymph nodes to say about that. Not that I'm aware of, anyway.
That's very interesting. And the second question is, so you mentioned it, but I missed it. What is the primary factor for increasing in volume of the lymph node?
No, you didn't miss it. No one knows that either. So you can see within 24 hours, both the weight and the volume have gone up tremendously in these nodes, and that's typical. You see this in lots of animals. I think the initial swelling is due to more fluid coming from the blood vessels into the lymph compartment. There's also the fibroreticular cells that Professor Bocharov mentioned. Those act like smooth muscle cells. They can relax and allow the lymph node to expand. There are other cell types in the lymph node that also have a latch state, like smooth muscle cells that you know well. And so those relax and allow the lymph node to expand. And then I think it's fluid flow coming from the blood vessels into the lymph node that makes that initial swelling. And then cell numbers go up as well. So we've done, I'm not showing the data here, but we've counted all the cells inside here. And certainly the cell numbers go up, but there's no way that cell numbers can increase by that much within 24 hours. So I think it's a combination of fluid flow, the cell relaxation, allowing the structure to expand, and then also eventually an increase in cell number for sure.
Yeah, it must be mechanical. But
we'd love to know more about that. So.
Can it affect the conformation of the porous compartments because they can effectively compress?
Yeah, it almost certainly does change the hydraulic conductivity of the cortex. But that, again, that's never been measured as far as I know.
Well, thank you very much.
Thank you.
Please, other questions, if there are.
Ask one more, question.
Yes, please.
One more, question. Very, controversial in some sense. James, what do you think of the possibility of the pumping function of a lymph node? Can it really work like? Can it? There are certain data showing that it actually, also could contract with a certain frequency. So, what is your view on this?
Yeah, I've seen lymph nodes show signs of contractility. My biological collaborators have described it. I would never exclude the possibility. Philosophically speaking, it's hard to prove something doesn't exist. So I would say it's a possibility, but I don't see it contributing so much to the flow. The vessels in, for example, the arm that approach the lymph node here under your arm, there are about 30 or 40 lymph nodes there. Those vessels are under 1 millimeter in diameter, and the vessels that lead out of them are under 1 millimeter in diameter, and they can only generate so much pressure with their contractions. And I think that's why the lymph node maintains this low resistance pathway around the outside, is not to restrict flow generated by those little vessels. But I haven't seen evidence of valve structures inside the lymph node. So if the lymph node contracts, basically it's just going to squeeze fluid sort of, out both directions. And I'm not sure what good that can do. So I've yet to really understand or see good evidence that a lymph node can contribute substantively to pumping, but it wouldn't surprise me, I guess. It's just not something we've looked at.
Okay, thank you very much.
Yeah, that's a good question.
Anastasia, now you can ask all your questions.
I even don't know what to ask.
Just remember what they ask you after your during your dissertation defense. You had many questions about lymph flow.
Forget about everything. But I also would like to ask about pumping lymph nodes, but Professor Bocharov has already done it. There are some physiological papers where lymph nodes are where it is shown that lymph nodes are pumping, and their conclusion is that they can contribute to the total lymph flow in the lymphatic system.
All right. Excellent. Well, I look forward to seeing that work. Excellent.
Yeah, but yeah, go ahead.
This work, the work that I know that is in Russian, but maybe we can find analogical work in English. I can send you if you're interested.
I would love to see it. I'd love to see it. Yeah.
But I should also say that Anastasia developed a model of the lymphatic system with just this huge system and modeling this using one-dimensional approximation. Probably a little bit similar as you modeled this lymphatic system, all these vessels inside the lymph node. So her work was this global lymphatic system. Yeah, and also what was the mystery for me, how, and you mentioned it at the beginning, how it can work against, well, if there is a pressure gradient or it works without pressure gradient, so how it works, in fact, can you answer this question? Because well, in our body there is this difference, like on the top, on the bottom, there is pressure difference. So can you comment on that?
So
whether pressure difference is necessary for this.
Yeah, well, I mean, if all fluid flow occurs due to pressure difference, so how do you generate it? I think is the key in lymphatics, and all of the pumping work that we've done, mainly with lump parameter models, which are kind of 0-dimensional or maybe half-dimensional, you might say. So what we found is that squeezing the vessel is actually very easy. That can occur due to the lymphatic muscle cells or due to external tissue movement. So squeezing the vessel is very easy. I think the key to lymphatic pumping is actually the opening up of the lymphatic vessel after it has squeezed. And that only happens if you have positive transmural pressure or some external tissue tethering the vessel to pull it back open after it has contracted. So every time, you can vary the pressure to relatively high values, and the lymphatic vessel can still do some squeezing, can still do some pumping. But if you compress it from the outside and don't allow it to expand after its contraction, then it will not pump anything. And I think that's the underappreciated part. So it's not the squeezing, it's the relaxation after.
Okay, very interesting. Okay, thank you very much. Other questions? If not, then I would, well, as you suggested, if you can send us your papers, for example, to Anastasia, so we will be very much interested to read them in more detail, and eventually, maybe if you allow, to contact you later about maybe if there are some questions.
Absolutely. Yeah, please.
Okay. Then thank you very much again for this very interesting presentation. And as we discussed, we look forward to see you soon in Moscow.
I hope so. Yes. Thank you all. It's been a great honor.
Yeah, thanks.
Thank you.
Thank you very much, James. Thank you.
Good to see you.
Thank you very much. Thanks to everybody. It's all
Bye-bye.
for today for our seminar, and we will meet next week. Okay, thanks. Bye.
Thank you.
Thank you very much.
Bye.